There are four clusters, cluster 1, 2, 3, and 4. So that's why it's called the homotetrameric enzyme, which as you can see over here in a ribbon shape. So zooming into each of the clusters, you can see there are three different colors. And each of these colors represent the secondary structure. So secondary structures are made from the primary sequence. To break this down even further, so you can think of like the primary sequence as like those adjacent amino acids like linked together to form like a linear line. So these like primary sequence, a lot of them come together. They start folding and interacting with one another to form secondary structure, which I have like mentioned here earlier. So the red is representing the alpha helix, yellow represent beta pleated sheets, and the green is representing the random coils. And as you rotate them around and start counting, you can see like there are 10 beta strands, and then helices and 3 random coils can be counted. So these structures go hand in hand together to form structural regions with specialized functions, including the regulatory domains over here, catalytic domain over here, and oligomerization domain around this area. So to change the color a little bit to make it easier for visualization, as you rotate around, you can see this like each of the cluster contains this bubble looking thing. So this bubble looking thing is called the BH4 and is found in the catalytic domain. And it is a cofactor, which is a non-protein component that helps PAH enzyme to break down phenylalanine. So most importantly, it allows the folding of the enzyme to be done correctly, and it has the chaperone-like effect in special terms in science. So it prevents the enzyme to be broken down, or in other words, degraded by a pathway called the ubiquitination